My name is Kai Como. I am a senior at Prairie View A&M University. I am a biology major with a chemistry minor, and I'm on the pre-med track. This summer, I was at Stanford University as an intern via the SenNet CUSP program with the support of the SenNet Consortium and the NIH. Today, I will be talking about a new imaging tool we worked on for arthritis imaging, which is focused on cellular senescence. The objective of this research is to test a new imaging biomarker that will detect senescent cells in musculoskeletal tissues. In the 3 months that I was at Stanford, I wrote a review paper on the expression of β-galactosidase in various tissues and organs during normal aging and pathological senescence. Throughout the presentation, I will be able to present an overview of the review paper as well as some research that I was able to shadow. So first, I want to go over some key terms that are the focus of this research. And these concepts are what I studied and the main focus of my research review paper. First, senescence. So cellular senescence is a state of permanent cell cycle arrest. Senescent cells are very unique because they will eventually stop multiplying, but they do not die. So this can cause a buildup of the cells that release chemicals that can later lead to age-related disease and conditions. Senescence can be induced by telomere shortening, genomic damage, strong mitogen-associated signals, epigenomic damage, and activation of tumor suppressors. Part of the motive behind this research is that there aren't enough imaging tools to detect senescent cells. Osteoarthritis is a degenerative joint disease that primarily affects the cartilage. That is the protective tissue that covers the ends of bones in a joint. So the cartilage gradually breaks down and wears away, leading to cartilage degeneration, bone growth, inflammation, and joint symptoms. Osteoarthritis is a common form of arthritis and is most commonly developed in people as they age. Beta-galactosidase is an enzyme that breaks down lactose to produce galactose and glucose to enter glycolysis. Beta-galactosidase is an important biomarker used to detect the presence of senescent cells and was also the main focus of my research paper. Now coming into the research project, we focused on the 18F PY-gal radiotracer. This radiotracer is a PY-gal substrate, which is hydrophilic, and it is conjugated with the 18F. When the 18F PY-gal radiotracer enters the senescent cells, it is cleaved by the beta-galactosidase, and this cleave signal is detected by the PET scan. In this study, we are testing the 18F PY-gal radiotracer in 2 different ways, in vitro and in vivo, which is using the large animal model. First, in the in vitro part, we will evaluate if the radiotracer can detect senescence using mesenchymal stem cells. And then we will move on to the large animal model, which are pigs. How do we induce senescent cells and mesenchymal stem cells? So first, mesenchymal stem cells are found in bone marrow and are important for making and repairing skeletal tissues. So these mesenchymal stem cells were isolated from the bone marrow of the pigs. Then they were cultured in the lab. So during these experiments, we seeded about 200,000 cells in a 24-well plate. Then these cells were incubated at 37 degrees Celsius with carbon dioxide, and the media was replenished every 2 days. When the cells obtain 90% confluence, that's when the cells are treated with 400 nanometers of doxorubicin on day 1 and day 3, and then incubated. So usually senescent cells were attained by day 5. And then the most important test performed was the beta-galactosidase assay. Here, these pictures show that the senescent cells are less metabolically active than the control cells. So in the first picture, the control cells have taken up nutrients from the media, and in the second picture, the senescent cells have not taken up nutrients from the media, and there's no change in the color of the media. So knowing this, we were able to determine that the senescent cells are less metabolically active. Beta-galactosidase assay is one of the most traditional assay forms of conducting senescence. As we see here, mesenchymal stem cells treated with doxorubicin have an increased expression of beta-galactosidase compared to the control, where we don't see much expression at all. For the imaging, we did PET-CT with the 18F PY-gal radiotracer, where we induced senescence in the confluent mesenchymal stem cells. Both control and senescent cells were treated with 20 microcuries of radioactivity and incubated for 1 hour. Then we obtained the PET-CT images. Okay, these are the PET-CT images where we can see that the range of uptake is significantly higher in the senescent cells than in the control cells. And this data was all quantified by nanocurie CC. As we concluded the in vitro portion of the project, we moved on to the in vivo part. This is involving the pig. This is the portion with the large animal model. This is where the project gets complicated and involves a number of multidisciplinary team, including the anesthesiologists and the surgeons. I was able to shadow several of these surgeries during my 3 months at Stanford. And as you can see, these are the surgeons who are operating on the pig. They open up the cartilage and create two defects. The first defect is filled with the control cells, and the second defect is filled with the senescent cells. Then the last picture is when they close back up the pig's knee. On day 2, the PET and MRI scans are conducted of the intra-articular injection of the radiotracer. If you take a look at the picture that has the senescent and the control labeled, we can see that the senescent cells have more of an uptake of the radiotracer than the control group. In conclusion, we were able to conclude that 18F PY-gal radiotracer can detect the overexpression of beta-galactosidase in senescent cells. In addition to the radiotracer was also able to track senescent cells in cartilage defects of pigs, which refers to the in vivo model. Lastly, I would like to give a huge thanks to the SenNet CUSP program and the NIH for creating this internship. I have had an amazing experience and have learned so much. I also want to thank Stanford University, Dr. Heike, Dr. Vidyani, and everyone in the Heike lab that I was able to work with during my time. I have learned so much about molecular imaging techniques, senescent cells, culturing cells, and going through this process of conducting research has been very eye-opening, and I am greatly appreciative of this opportunity.